Good afternoon, everyone, and welcome back to the Popular Science Talk session. It is my pleasure to introduce our second speaker, Dr. Maanasa Natrajan. She obtained her PhD in neuroscience from Johns Hopkins University and is currently a Caliber Fellow at ICTS. Dr. Natrajan is a computational neuroscientist whose work combines theory and modeling with data analysis. Her research aims to understand how the brain learns continually, stabilizes memories, and extracts generalizable structure. The title of her talk is The Brain's GPS, How We Build Mental Gaps. Let us welcome Dr. Natrajan.
Hello, everyone. Thank you so much for having me here. I want to share with you all a story about the brain's GPS. So how is it that we build mental maps of the world around us? So let's start by thinking about a person going along a trail. Sorry. Is that better? Or does it become, okay. So yeah, let's think of a person kind of going along a trail in a forest or in the woods. Now, there may be multiple sections in the trail where there are tall trees, so it all appears pretty much identical. So if you haven't been to this trail before, you might get confused about where you are in this environment. However, if someone is familiar with this trail, they've been here many times, then they'll know that even though these A and B kind of like look very much the same, they actually are distinct locations. And that from A, if you turn right, that leads to a campsite. And then from B, continuing straight leads to a waterfalls. Now, these campsite and waterfalls themselves might not be kind of like specific experiences. For instance, in this trail, there might be multiple waterfalls and multiple campsites, suggesting that they might also be sensorily ambiguous. So then, how is it that we're able to kind of infer in this environment where exactly we are and what actions should we take to reach specific goal locations? Well, it's perhaps because we have an internal map of this external environment, or a mental map, or in neuroscience, this is known as a cognitive map. Now, to understand cognitive maps, we might want to first look inside the brain and see how is it represented, what is happening inside. So you could perhaps look inside the brains of humans, but what turns out easier are these rodent models. So mice and rats, you can let them kind of explore some spatial environment, and then you can record neural activity from their brains. So over here is a rat kind of running along this square environment. And these red dots indicate when a particular neuron is active or is spiking. You'll see that as the animal kind of like explores this particular neuron seems to like to fire when the animal is in this region of the space, and not so much in other regions of the space. Now, I guess a day ago, you heard a talk from Sarthak where he showed you kind of maps which had kind of hexagonal lattice. This is from a different brain region where the cells fire specifically in a particular location or place within the environment. And then we can average out kind of over space to get what is known as a firing rate. So over here, the blue indicates kind of low firing rate. So this neuron is not firing at those locations. And then red indicates much higher firing rate. So this particular neuron likes to fire at this particular location or place. And so neuroscientists named it a place cell. Now, this is for a particular cell. If you look at many different cells within this brain region called the hippocampus, different cells will have kind of fields at different locations. So they may fire at different locations within the environment, and together they tile that entire space, suggesting that these cells might be together forming a spatial internal map of this environment. Okay. So these internal maps are not just limited to kind of such spatial environments, but they can also code for much more complex task environments where you need more context. So what neuroscientists did was to kind of let mice or rats run around in this like weird shaped environment where it starts over here, kind of at this top right corner, runs down, and then along this like common loop, and then to the left. And then another trajectory, they would make the animal run starting from this left, down, and again along this common loop, and then to the right. So there are two different trajectories. What you'll see is that the animal passes through this common arm along both these trajectories, in which case the sensory observations that the animal might be getting would be very much the same, yet from which direction it came and which direction it needs to move would be different. So these are kind of like states where the history and the future trajectories are going to be different, even though it's the same physical space. And what they did was to record neural activity from similar cells in the hippocampus. And over here is the firing rate or the activity of a neuron in this location under these two different trajectories, the blue trajectory over here and the yellow trajectory over here. What you'll see is that this neuron tends to fire a lot kind of within when the animal takes this blue trajectory and passes through the space, but not when it passes through the space along the yellow trajectory, suggesting that it's coding for this location in a different manner depending on the historical context and what the animal is going to do in the future. So since it kind of splits between the two, they were called splitter cells. Similarly, they also looked at other kind of like tasks. In this case, the animal had to run along this environment 4 times before it would receive a reward. So it just goes 1, 2, 3, and 4, and then the 4th time it goes over here, it gets a reward. And then it has to repeat the same thing again. And then what they did was to look at the neural activity. So these are all of the neurons that they recorded from, along the 4 different labs across kind of different positions. And over here, again, the darker red color indicates much more higher firing rate. What you'll see here, some of these neurons, they tend to fire in lap 1 at specific locations, much more than they do in lap 2, 3, or 4. And similarly, there are a different group of cells that tend to fire much more in lap 2, others that are more specific to lap 3 or lap 4. And together, you'll see that they don't just encode that spatial environment, but now encode this entire kind of task environment, which consists of these 4 different labs. So in turn, what this suggests is these cells aren't just coding for something based on the current sensory observation, but are actually, in some sense, coding for some context-dependent map that is relevant to the animal in the specific task or for that specific world. Now, these hippocampal cells have been very well studied after animals have learned these internal maps or cognitive maps. But what is less known is how are these maps formed? Like, how do these cells gain these specific firing properties? So the main question that we had was, how do neural response patterns change as animals gradually learn and form an internal map of a new environment? Now, in addition to the experimental side of things, over the years, there have been lots of different models of cognitive maps as well. The key idea here is that perhaps the world has some structure in it, and it generates specific sensory observations. So as the animals kind of navigate along this space, for instance, let's say the animal starts over here and then takes an action to the right, then the animal would see a book, or the world would show a sensory observation of a book, and then it might go down, in which case it would see a sensory observation of a table, left, and so on. And the idea is that perhaps you want to learn a map of this environment. And one class of models suggested that perhaps to learn this, what you might do is to try to predict what observation is going to come next. And then you'll get the sensory observation. Now you can perhaps use that to kind of error correct, and in turn, kind of perhaps that might lead to formation of these internal maps. So based on kind of next time step prediction. Another class of models directly tries to abstract out this particular task state representation from the world. So these Z1, Z2, and so on are kind of the states of the world, and A tells you what action. So let's say you start at Z1 and take specific action A1, then that leads to the state Z2, and so on. And these X's are the sensory observations. So then in this case, the idea is to learn how states transition from one to the next and what sensory observations they generate. Now, these different classes of models that have been suggested for cognitive maps have been able to replicate many aspects of the hippocampal neuron firing. For instance, they can generate, so these models can generate things like these place cells, So over here, this is now an artificial neuron that selectively fires at this particular location within the environment. They can also generate other things like lap cells. So when you have kind of 4 different laps before which you get a reward, there are cells that specifically fire at a particular location in lap 1, but not in the others. Cell that fires more in lap 2 when compared to the others, so on. So many of these models have been able to replicate different aspects of kind of these fully formed cognitive maps. Our main question is, what about the learning process itself, right? Can we model the learning dynamics of cognitive map formation with some class of these models? Okay, so these were the 2 main questions, right? How do the neural response patterns change as animals learn cognitive maps of new environments? And how can we perhaps model these learning-related dynamics? This is a work that I did during my PhD with my PhD advisor, James Fitzgerald. And this is a collaboration with Nelson Spruston's lab, who's an experimentalist. Weinan San and Johan Winnebust led all of the experiments that I'm going to be describing now. And I worked more on the modeling and the data analysis aspects of the project. Okay. So what do? They let a mouse kind of sit on a ball and then run on it while it was being surrounded by monitors that show specific scenes or sensory observations. And as the animal would move, then these sensory observations would be kind of changed such that it appears to the mouse as if it's moving through space. So this is a virtual reality environment, kind of like, in some sense, the mouse playing a video game, say. And then the animal would have access to this water port where basically at specific location, it'll get rewarded. And if the animal licks over there, then it'll get a reward. So the experimenters trained the animal on this particular virtual reality environment, which consisted of 2 different tracks. You'll see that they are almost identical except this indicator region. The stripes indicator suggests that the animals will get rewarded in this near reward zone. And in case of the circles indicator, the animals get rewarded in this far reward zone. The key idea here is, again, that you have these gray sensory observations that are repeated multiple times kind of within a track, as well as between the 2 tracks. Again, creating sensory ambiguities, just like in the trail example, there were tall trees that all looked identical. And then if you look at the reward zones, the near and far reward zones in both the trials, they look pretty much the same. Yet in one case, the animal needs to lick here, and the other, the animal needs to lick there. So the idea was that perhaps to perform well in this task, the animal would have to maintain information about this indicator and in turn lick correctly. And thus the idea that perhaps it can't just use the current sensory stimulus, but needs to build a map of this task space or environment in order to perhaps do well in the task. Now, as animals kind of like they run through this track, at the end, the animals are teleported back to the beginning. And then the next trial can be near or far with equal probability. So then it's like a continuous stream. They're running through the track, get teleported back, and then the next one could be near or far. And then, yeah, they lick and go on. Okay, so very early on, the animals don't have a good sense of where they'll be rewarded. So what they do is lick everywhere. So over here, I've divided kind of the data from near and far track into 2 separate plots. Along the x-axis are different positions along the near track, along the far track. And on the y, we're going to see how this behavior changes over time during learning. So the top is basically very early in learning. This is when the animals have no sense of where they'll be rewarded. And each of these dots indicates a lick of the animal. You'll see they lick pretty much everywhere along both the near and the far tracks. Right? They don't know anything about this environment. Now, over time, with more and more experience, they realize that they don't get rewarded in this beginning of the track at all. So they stop licking there. They lick in this near reward zone in the near track. They get a reward here, so then they stop licking. In the far track, they again lick in this near reward zone. They don't get a reward here because this is a far track. And they continue to lick until the far reward zone, where they'll get a reward, and then they stop licking. And then over more days of training, the animals actually realize kind of the association between the indicator and the reward zones. And they selectively lick only in this near reward zone in the near track and far reward zone in the far track. So the behavior changes kind of sequentially over time. And over time, they learn kind of this exact association. So over here, I'm going to show you the track and perhaps like a well-performing animal. So here you'll see the circles indicator, which means it's a far track. The animal is running through. This is the near reward zone. It doesn't lick here. It goes till the far reward zone. You'll see that it starts licking, stops, gets the reward, and then kind of continues after some time to the end of the track. And then the animals are teleported back to the beginning. Now, this is a stripes indicator. So it is a near track. Now, in this case, you'll see that the animals kind of lick in this near reward zone. Over here, you can see the animal licking, and then the animal gets a reward. And then after receiving a reward, it kind of runs through and runs beyond this far reward zone without actually licking through. So indeed, the animal has learned to lick correctly in the rewarded locations. Now, as animals learn kind of this task and perhaps the environment, we can ask what is happening to the neural activity. So what we can perhaps do these days is that you can genetically modify these animals such that whenever the neurons are active, they tend to glow, which means what you can do is put a microscope and then just and then look at the activity. So each of these flickerings that you see is kind of a neuron becoming active and inactive. And you'll see thousands of neurons in this region. And you can record from all of these neurons over time as animals are still learning the environment. So we can ask, how is the neural representation actually changing? So the next thing we do is we segment these neurons. So we track each of these over time. And then we end up with a very large matrix of neurons by time. And then the question becomes, how do we make sense of this very high dimensional space of neural activity patterns? One simple thing that we can perhaps do, so over here is the near track along the x-axis at different positions. And I have all of the neurons along the y-axis. Now, the activity of all of the neurons at a particular position or particular state in this track can constitute a population vector. So you can think about a space where kind of each of the axes is an individual neuron or its activity. Then this is simply a population vector within this kind of higher dimensional space. Similarly, the activity of all of these neurons in this region kind of after the near reward zone will constitute another population vector corresponding to that. And now we can ask how similar or different are these neural representations of these distinct locations. Now, if they're very similar, so what we can do is perhaps take a dot product or a correlation, right? If they're very similar, the angle between them will be very small, and the correlation would be very large. Or perhaps they might be very different from each other, as in this means something very different to the animal and in the world. In this case, they would be orthogonal. So now we can compute these population vector correlations. So this is a population vector correlation map. On the x-axis are different far trial positions, and on the y, different near trial positions. And the color here indicates the value of the correlation between these population vectors. So let's go step by step and look at the population vector correlation between this beginning of the track gray region and this gray region after the near reward zone. Okay? So which is beginning of the track gray region and this gray region after the near reward zone over here in this like white square. And you'll see that this color is kind of light, more reddish or light, which indicates that these representations are quite similar to each other. Right? The correlation is closer to 1. And overall, you'll see that every time the sensory observation is gray, the neural representation appears to be correlated. Every time the sensory observation is kind of this near reward zone, the representations are correlated. The representations for kind of these far reward zones in both the trials are correlated. So basically, it's the same sensory observation. The neural representation appears to be very similar. Now, we can ask, what about the neural representation when the current sensory observations are very different? So in this case, the beginning of the track gray region and the stripes indicator. So beginning of the track gray region and the stripes indicator. You'll see that the color is pretty much black, which is like a zero correlation, suggesting that these neural representations are almost orthogonal to each other or very different from each other. And you'll see kind of like every time the sensory observation is different, the representation appears to be different. So very early on in learning, it appears that the animal just has one state corresponding to the gray sensory observation. And then when it sees an indicator, either the near or the far track indicator, it diverges, but then returns back to this gray observation state when it again says gray. So it's confused, it doesn't know that it's actually progressing kind of through this track environment. It thinks that it's returning kind of to the same location, at least based on the neural representation. So early on, the neural representation seems to match that of the current sensory observation. But let's see what happens over time after some learning. The animal then seems to learn that the same gray observation is repeated multiple times within a track, and these representations become decorrelated, right? They're much more black, or now they're much more orthogonal to each other. So it's learned the linear structure of the task. However, you'll see that it treats both the near and the far tracks in pretty much the same way. This diagonal is pretty much correlated, except at this indicator, which is basically different sensory stimuli. So the animal goes through kind of like this stage where it realizes that there's some linear structure, but doesn't exactly know that they are kind of two different tracks. And this is a phase where the animal would basically lick in both near and far reward zones. And then the next stage, based on whether or not a reward is received in this near reward zone, the representation of this post the first reward zone region, that tends to decorrelate between the near and the far tracks. And behaviorally, this is kind of the phase where the animal would lick in the near reward zone in both near and far, and in near it would get a reward, so that representation becomes different, and it wouldn't lick in the far one. And in case of the far track, it wouldn't get a reward, and its representation is different, it produces a different behavior. And then over time, the animal actually learns this entire structure of the task. So orthogonal representations all the way from the indicator to the second reward zone R2. Thus representing kind of the task states or the states of the world or the environment with these orthogonal or very different representations. So what we found is that during this process of internal map formation, started off with representing current sensory stimulus to then go through a sequence of decorrelations such that it ended up with representing the map, the model of the world with these orthogonal representations. Okay, so now we can ask, how might we model this process? So the animals over here actually experienced a continuous stream of sensory observations, but perhaps we can think about that, think about discretizing it. So at some time point t, it receives some observation xt. In this case, it's the stripes, xt plus 1, again stripes, and then the gray region, and so on. We can also think about the world as being composed of discrete states. So as the animal kind of like runs through the environment, perhaps transitions from one world state to another world state. Now, let's say that let's denote this state at time t as zt. We define the world state based on two pieces of information. One, which track type it is and how far has it traveled from the beginning of the track. Then, this next state at time t plus 1 will only depend on the state at time t and not all of the previous states. Right? Once you know that you are at 100 centimeters and near track, you know that you're going to be at 120 centimeters and near at the next trial. You don't need to know that you were at 100 centimeters and near or 90 and near before. So this is known as a Markovian property. And essentially, the animal, as it passes through this environment, it can learn to predict what is going to come next by just using its current state if it had access to these world states. However, in reality, the animal doesn't exactly get this information. right? No one is telling it that, okay, you have traveled 110 centimeters and you are in the near track. What it gets are sensory observations, which are ambiguous, right? Different world states might generate the same sensory observation. So the world states kind of in this like post-indicator region also generate gray observations, post-reward 1 region also generate gray observations, and so on. So perhaps what the animals need to do is to build a world model or an internal map by first trying to infer what world states they are in, how they transition from one to the next, and what sensory observations they generate. So essentially, if given that at time t, it's in some state zt, what's the probability of transitioning to some other state zt plus 1 at time t plus 1? Right? Those are the transition probabilities from one state to the next. And then what are known as emission probabilities, which is telling you what is the probability of seeing this observation xt, given that you're currently in this state zt at time t. So essentially, all the animal is getting are sequences of sensory observations. The idea is that perhaps it needs to kind of like modify these probabilities in order to maximize the observed sensory sequence x1 to x3. Now, there's a particular class of hidden Markov model known as clone-structured cognitive graph, which was shown to replicate many aspects of the fully formed cognitive maps. So in this clone-structured cognitive graph, there are multiple clones or states corresponding to a particular observation. What that means, all of these different, so irrespective of which of these states you're in, the probability of seeing observation gray is always 1, and the probability of observing any other sensory observation is 0. Right? For every observation, there are some given number of states. So in some sense, this emission probability now becomes fixed and deterministic. So once you know that you have observed the state, you know that that is going to generate a sensory observation of reward with certainty, and that it's not going to generate any of these other observations. So then what you're left with are just the transition probabilities, probabilities of going from some state at time t to some other state at time t plus 1. And the idea is that perhaps the model would learn to update these transition probabilities to maximize, again, the probability of the observed sequence. Now, what might this model be able to do? Because there are multiple kind of clones or states corresponding to a particular observation, it can learn to use different clones or states for, say, the beginning of the track gray region, the gray region after the indicator in the near track, gray region after this reward zone 1, after this reward zone 2, and so on. So by having this clone structure, you might be able to kind of use these different clones or states to correspond to different world states that all generate the same sensory observation. Now, this happened to have this kind of like clone structure, which turns out to be useful if you have the same kind of like world model, but like different sensory observations. Then by learning such a model, once you learn the transition structure, you can kind of quickly map on to other sensory observations by, say, learning the emission probabilities. So it provides a flexible way of like learning some structure, which in turn might be common across environments, which is not going to be true here, but we'll see that, but this could be helpful for the animal in real world environments. Okay, now one might ask, this seems like an abstract model. How are we going to relate this to the neural representations that we looked at before? One way we'll do that is to think about each of these clones or states as being similar to a neuron. The hypothesis is that perhaps the probability of being in a particular state is similar to the firing rate of a neuron. Then, just like we had population vectors in the neural example, now we can have state probability vectors. So having experienced some number of sensory observations, what is the state probability of being in each of these states? That will correspond to now a state probability vector. And then we can do a similar thing where we correlate these state probability vectors like we did for the population vectors. Okay, so along the top are going to be the correlation maps that you saw for the animals before. And at the bottom would be the correlation maps now for the state probability vectors for this hidden Markov model. Now, very early on, so before these transition probabilities have been updated in any way, you'll see that every time the sensory observation is the same, the state probability vectors are also the same or very correlated. And that's because now everything is being dominated just by the emission probabilities. There's nothing that has been learned about the transition matrix. And then we let this model to experience a similar set of sensory observation sequences as did the animal, again drawn from that same world. Over time, we find that just like the case of the animals, this model also learns the linear structure of the task. So it decorrelates kind of these state probability. It uses kind of different now states for distinct locations within a track, but continues to use the same set of states to represent the two different track types. And then, just like in the case of the animals, the model also learned to decorrelate this region after the near reward zone between the near and the far tracks. Again, just like what the animals did based on whether or not a reward is received. And finally, they also decorrelated this kind of region after the indicator, this gray region after the indicator based on the indicator, thus drawing kind of that association. And forming orthogonal representations all the way from the indicator to the second reward zone between the near and the far tracks. So this simple hidden Markov model, which just used a set of sensory sequences without using any specific reward information. The reward was just used as like a sensory observation rather than anything like reinforcement learning or where the reward itself was playing some other additional function. Without that, this model was able to replicate not just the final orthogonal map as that of the animals, but also the specific learning trajectory. Now, in case of animals, we wouldn't know why exactly it followed the specific trajectory, but with this hidden Markov model, we can actually understand kind of why this particular trajectory occurred. But I guess one question that one might ask is, okay, so this model was able to replicate the final map and the learning trajectory. How novel is that? Like, let's say we use some other model, would that also be able to replicate this final and the learning trajectory? So another class of commonly used models that I mentioned before was based on next time step sensory prediction. So over here, we'll compare this to a neural network model. So what does a neural network do? It takes in some inputs, performs some linear and then some nonlinear operation in it, and then throws out some output. The idea here is that given the sensory observation at time t, the network needs to generate the observation at time t plus 1. Like what is going to come next? Now we'll use a recurrent neural network. What does that mean? In this layer, the activity at time t minus 1, so yt minus 1, will in turn feedback onto and influence the activity at time t. So that's a recurrent network. If you didn't have this connection, that would be a feed-forward network. Now the reason we want a recurrent network in this setting is let's say it was simple feed-forward network, then you get a gray observation, whether that is irrespective of in which sequence it is, right? The model would simply generate the same output. We want to maintain information about the sequence or the context history, so recurrence is kind of crucial. Okay, so then let's consider two different time points, T1 and T2. And now in here, these weights, these recurrent and feed-forward weights would be updated so as to ensure that the system generates the correct next time step prediction. Very common, very similar to kind of like commonly used machine learning and AI algorithms that are based on like either masked token prediction, all of these like large language models and so on. So let's consider a case kind of a time T1. The system observes observation gray. That leads to some pattern of perhaps neural activity, YT1, in some high dimensional space. We can think about this readout as some low dimensional projection of this higher dimensional neural activity. So this yellow space is the readout relevant space. And this is the more higher dimensional space. These dimensions are irrelevant to the readout. And now at time T2, the system again observes the same sensory observation gray. It leads to some other pattern of neural activity YT2. And now it needs to generate a different sensory observation for the future time step. And in this case, if you think about like a one-hot encoding, right, where each of these are completely orthogonal observations, then that would mean that this needs to be orthogonal. The output needs to be orthogonal. However, the neural activities themselves, the representation at time t1 and t2 themselves don't need to be orthogonal. You can have correlations as long as these are along the space irrelevant to this readout. So you can have kind of correlated representations which generate kind of this correct orthogonal readouts. What that means is perhaps the animals didn't exactly have to decorrelate the representations. They could have made small differences that could have been read out by a readout layer, but they didn't do so. However, if you train the most commonly used kind of machine learning algorithms, networks, so like simple recurrent neural networks, then they can very easily solve this task. They'll be able to predict the next time step observation. However, if you plot similar population vector correlation maps, you'll see that even for very different kind of states, they maintain highly correlated neural representations. And that's because they don't have to actually orthogonalize them to do the specific task, which is next time step prediction. Right? All they need to do is generate these correct readouts, which might be orthogonal, but these representations are much higher dimensional and they could be highly correlated. And this is for a simple recurrent neural network model. We kind of see similar things even for transformers. These are again kind of the models the model architecture that's on the basis of large language models. But then we can ask, why did the animals actually decorrelate them? Is it actually a better solution? Is it beneficial for something? So let's say that you start off with representing two distinct world states with highly correlated representations, and there's some noise in the system. Then you might end up with pretty much aligned representations, such that there's confusion between the two different locations. In contrast, by orthogonalizing the representations, now even if you have some noise, the representations after adding noise still remain very different, in the sense that they are going to be much more robust to noise. Now, animals often have to kind of do long-term planning, imagine trajectories of actions they may take and what observations they'll get. With all of that, you'll add more and more noise, and that adds up. So by orthogonalizing these representations, the animals learned to represent things that are in the world different with orthogonal or distinct representations, even though they provided the same sensory observation, suggesting that the kind of solution that the animals found, and perhaps what we saw with the hidden Markov models, was much more robust. So then what we could do is we took these insights and tried to mimic a hidden Markov model in a recurrent neural network. So we tried to, again, do the same thing for each neuron. Now, for each state in the hidden Markov model would correspond to a particular neuron, and then added in some constraints, like about what the feed-forward and recurrent weights would have to be to satisfy kind of like a transition and emission probability-like nature, and then applied a particular activation function, like a softmax, which basically takes in small differences and maximizes them. We found that such a hidden Markov model captured this orthogonal representation structure, that of the animals and that of the hidden Markov models. And again, this would be much more robust to noise and some long-term planning than a model which was just trying to do kind of next time step prediction without some of these architectural constraints. So all of these deep networks can always find solutions, but then the question becomes, will they find good solutions? And what is good can be determined based on different things. In this case, based on the robustness of that solution. And perhaps you can introduce specific biases into the architecture that can help them find those much more robust solutions. And perhaps thinks about the biology and thinks about, and these simple mathematical models can help us provide insights into what architectural constraints to actually impose. Okay. So till now, I've talked mostly about this virtual reality environment. This perhaps extends to any space. You might think about yourself walking along a road, and there may be kind of lots of chains, say a McDonald's that's in every block, or some other different buildings that look pretty much identical. Yet we are able to kind of build maps of these environments, and perhaps we represent them very differently because in the physical space, in the real world, they actually correspond to distinct locations. But this doesn't even have to be limited to space. Let's consider this sentence. As a cricketer picked up his bat, a bat flew overhead. I'm sure that even though the word bat is kind of repeated twice in this sentence and means very distinct things, even despite it having the same kind of sensory observation, you probably thought of very different things for this bat and this bat. Right? You probably thought of a cricket bat and a bat, a mammal that flies. So again, these sensory ambiguities are very common in language as well. And what gives us information is the context. And perhaps we represent these two different bats in our brains with very different representations because they mean different things to us. Now that was about language. This is even more so true for music. With just 7 notes repeated in many different kind of sequences, there are lots and lots of songs. And we're able to, despite hearing the same sa or same re, because it occurs in different sequences, we're able to map them to different things, expect what's going to come next, know what came before. So this idea of building a map isn't just limited to physical space. And perhaps this idea of simple hidden Markov model learning, capturing kind of this phenomena, would mean that what the brain is doing, whether that be for physical space, or for music, or for language, or for just generally any other space, is that it's trying to extract the structure. There are things that are hidden or latent from the animals or humans, and they're trying to extract, infer what those states are, represent them with orthogonal distinct representations when they actually mean different things, and to learn how they transition from one to the next in sequences. Okay. So overall, we asked 2 main questions. How do neuronal response patterns change as animals learn cognitive maps of new environments? And then what we found was that early on, the representations, when you get the same sensory observation, they are very correlated. But it undergoes a sequence of decorrelations to learn kind of these orthogonal representation states, thus going from kind of a representation of the current sensory information to something about the world state, thus forming kind of an internal map. And then we asked, how can we model these learning-related dynamics? What we found was this hidden Markov model actually captured both the final map as that of the animals, as well as the specific learning trajectory of the animals, suggesting that perhaps we can think about learning structure in the brain as similar to kind of like a latent state inference problem in a hidden Markov model. Now with these, the main aspects that I want you to take away are that we can use mathematical tools to help us analyze data, whether that's coming from neuroscience or biology or elsewhere, and to perhaps build models of these phenomena, which help us better understand them. And finally, perhaps also to improve certain other, say, ML/AI algorithms. Yeah, with that, I think I'd like to thank you all for listening, and I'm happy to take any questions. Yeah. Yeah. Yeah, it's actually a really nice question. So these animals, I guess the idea is that in the near reward zone, they get a reward sooner. In the far reward zone, they get a reward later. And in some sense, this particular reward, in the hidden Markov model, we were able to kind of like characterize the learning trajectory and see that basically this reward happening here actually caused this decorrelation earlier in this region than kind of in this region. But more generally, so some of these animals, many of these animals go through the specific learning trajectory that I showed you, which takes a long time. So they start off by thinking that every time I see the same observation, I'm coming back to the same state, and then they learn by decorrelating. However, and this I think took an order of 7 to 8 days. Each of these kind of like dotted lines indicates like a day. So they took a few days of kind of like going along in this environment. However, there were some other animals which started off with a different, say, bias or prior, where every time they saw even the same observation, they just always represented things differently. These animals learned by correlating the representations of the same states rather than by trying to decorrelate. And these animals apparently learned way faster, so within a day or two. And that also suggests how differences in kind of your prior or initial system could influence the learning trajectory and the learning speed. And even though most of the animals learned kind of through this like decorrelation, there were few animals which kind of just, I guess, thought of it like an infinite track, where every time I see something new, I'm just moving forward, so it must be a different place. And then they learned that, okay, some of these places are not different, they're actually the same. So they learned by correlating things. We tried to play around with the hidden Markov model as well to try to replicate some of these things. Yeah.
So how is that? What type? Like why not a CNN?
Yeah. So convolutional neural networks are very useful when you're thinking about visual information, because in some sense, there are, and they are also kind of inspired by the neuroscience literature, where the idea is that in physical space, so things that are kind of like closer have this kind of like receptive fields where you either like, yeah, the convolution, the kernel itself is in some sense also inspired by biology. Over here, the main idea was that the network needs to maintain some information about the past in order to do good prediction, because the current sensory observation is not sufficient, which is why we decided to use a network with some recurrence and not like a feed-forward network. Convolutional network would be more useful if you're thinking about things where there's some 2D space. Yeah. I think a couple of things. So first, we wanted to understand what is the brain doing, which was through the decorrelation process. And then the next part was to try to understand, okay, how might we model this process? And that was the approach at the kind of more algorithmic level. It looked like hidden Markov model captured that well. And then the next part was actually, I didn't have the time to talk about it, but like was about trying to mechanistically, how is the brain doing it, right? In all of these, the hidden Markov model, it's trained with like an expectation maximization, where basically you're using information about the past, about the future, and so on to update these transition probabilities. But that's perhaps not how the brain might do it. So what we also then did was to try to mimic what the brain might do by having a recurrent network where the weights are updated just based on pre and post-synaptic firing. So the neuron, the weight from one neuron to another is just updated based on the activity of these two neurons. That's local information rather than kind of more like global information. Sorry? In the network. So the idea was in some sense to get a sense of what is the brain doing, how is it doing, perhaps at a algorithmic as well as a mechanistic level. And I think another, which wasn't initially the goal, but another thing that we went on to do was more think about what is it about, what is it that we can add to simple recurrent neural networks to achieve these kinds of robust solutions that they didn't automatically do. Yeah, I guess so. A lot of these are very over-parameterized models in the sense that you can do a lot of things with them and they can learn lots of things. But then the challenge becomes figuring out what constraints to put in to figure out so that they behave like the brain rather than like every other possibility. So a lot of, I think, neuroscience research now kind of focuses on trying to add specific constraints into these models. I think the question of like, do we have a network that behaves exactly like the brain? Sorry?
Some local function.
Yeah, I think for specific functions, so like there are things like working memory, where like basically you're trying to remember information remember information and like use them to change some things. For those, some of these recurrent neural network models perform very similar to what people have seen in humans and other animals. So like think about, I say a sequence of numbers, like a phone number, you can probably remember it. And that the way that happens seems to be very similar to how these RNNs do it. But then I guess a lot of questions remain about like, and in some sense, no model is gonna, the idea is to abstract away something. So like the idea isn't exactly to create another model, like another brain outside, which will function exactly like that. But in some sense, to be able to create, abstract off a model which you can then play around with and understand, which might be harder with the brain. So any model is not going to, in some sense, replicate exactly every specific thing, but then the question becomes, what are you interested in? Is it able to replicate that?
So as there are no more questions, we will conclude here and the speaker again for the one.
Thank you.
Dr. Natrajan is also available for interaction after the session.